DNA, the molecule that serves as the blueprint of life, is remarkably stable and precise in its function. However, it is not immune to changes. These changes, known as mutations, can occur naturally or as a result of external factors. Today we'll explore what changes in DNA look like, how they happen, and why they are significant to life, health, and evolution. Let's start with the basics. DNA, or deoxyribonucleic acid, is composed of smaller units called nucleotides. Each nucleotide consists of a sugar molecule, deoxyribose, a phosphate group, and one of four nitrogenous bases, adenine, A, thymine, T, cytosine, C, or guanine, G. These bases pair specifically, adenine with thymine and cytosine with guanine, forming the iconic double helix structure. The sequence of these bases forms the genetic code, which determines the traits of an organism. A single change in this sequence can have significant consequences depending on where and how the change occurs. Changes in DNA are referred to as mutations, and they come in several forms. One of the simplest types is a point mutation, where a single base is substituted for another. For instance, if an adenine, A, is replaced by a guanine, G, in a gene, it could change the meaning of the genetic code. Some point mutations have little to no effect, while others can alter the structure and function of a resulting protein, leading to observable traits or even diseases. Another type of mutation is an insertion or deletion, where one or more bases are added or removed from the DNA sequence. These mutations often cause a frame shift, changing the way the genetic code is read during protein synthesis. Frame shift mutations can significantly alter the resulting protein, often rendering it non-functional. Larger-scale mutations can involve whole sections of DNA. For example, a duplication mutation results in a segment of DNA being copied and repeated, while an inversion mutation occurs when a section of DNA is flipped in orientation. Chromosomal mutations, such as deletions or translocations, involve entire pieces of chromosomes being rearranged, which can disrupt multiple genes at once. What causes these changes in DNA? Mutations can arise from several sources. Spontaneous mutations occur naturally during DNA replication. While the process of copying DNA is incredibly accurate, occasional errors can slip through. Fortunately, cells have repair mechanisms to catch and fix most of these mistakes. Environmental factors can also cause mutations. Ultraviolet, UV radiation from the sun, for instance, can damage DNA by causing thymine bases to bond incorrectly, creating what's known as a thymine dimer. Similarly, exposure to certain chemicals like those found in cigarette smoke can chemically alter DNA. Even viruses can contribute to mutations by inserting their genetic material into a host cell's DNA. The significance of DNA changes depends on their location and impact. In some cases, mutations are neutral, causing no noticeable effect on the organism. This often happens when the change occurs in non-coding regions of DNA or doesn't alter the final protein. In other cases, mutations can be harmful. For example, a mutation in the gene responsible for producing hemoglobin, the protein that carries oxygen in the blood, can lead to sickle cell disease. This condition causes red blood cells to adopt an abnormal shape, leading to reduced oxygen delivery and other health complications. Mutations can also play a role in cancer. Changes in DNA that affect genes regulating the cell cycle, like proto-oncogenes or tumor suppressor genes, can lead to uncontrolled cell growth. For instance, mutations in the BRCA1 or BRCA2 genes significantly increase the risk of breast and ovarian cancer. Despite their potential for harm, mutations are not always negative. In fact, they are a driving force of evolution. Mutations introduce genetic variation, which is essential for natural selection. For example, a mutation that improves an organism's ability to survive in its environment can be passed on to future generations, becoming a beneficial adaptation. Consider the case of antibiotic resistance in bacteria. Mutations that allow bacteria to survive exposure to antibiotics can spread rapidly through a population, making the bacteria harder to treat. While this poses challenges in medicine, it's a striking example of how changes in DNA drive adaptation and survival. Understanding mutations also opens doors for advancements in science and medicine. Genetic testing can identify harmful mutations, allowing for early intervention or preventive measures. For example, individuals with mutations in the CFTR gene can be diagnosed with cystic fibrosis, a condition that affects the respiratory and digestive systems. Early diagnosis can improve management and outcomes. In addition, scientists are developing tools to correct DNA changes. One such tool is CRISPR-Cas9, a revolutionary gene editing technology. CRISPR allows scientists to precisely target and modify specific DNA sequences, potentially curing genetic diseases or enhancing desirable traits. To visualize these concepts, imagine DNA as a long, intricate book written in a four-letter alphabet, A, T, C, and G. A mutation is like a typo in this book. Some typos might go unnoticed, others could change the meaning of a sentence, and still others might rewrite an entire chapter. The significance of the typo depends on where it appears and how it alters the story. In conclusion, changes in DNA, mutations, are an inevitable part of life. While they can lead to diseases like cancer, they also fuel evolution and diversity in the natural world. By studying these changes, scientists can unlock new ways to diagnose, treat, and even prevent genetic disorders. The story of DNA is one of resilience and adaptability, with each mutation adding a new twist to the ongoing tale of life on Earth.